So our next speaker is Melissa Chiu from Seattle Cancer Care Alliance. Melissa is the recipient of our Technologist Award. Congratulations, and Ming will present the award. So Melissa will tell us about a comparative analysis of reimbursement for molecular pathology testing for neoplasia.
I'd like to first thank the meeting organizers for the award and the opportunity to present here, and thank you all for being here. So this one, okay. Oops. Okay. So last year at this conference, my colleague Kate Kroger presented the laboratory testing pathway that was implemented at Seattle Cancer Care Alliance. The Evaluate for Hematopoietic Disorder, which is shorthand as the EVAL-HD at our institution, is a standardized testing pathway that was developed by laboratory experts and leading providers from different specialty areas. Its primary goal was to offer consistent and efficient testing based on medical indication and to develop actionable results. For majority of subtypes of hematopoietic disorders, morphology, flow cytometry, and karyotype will be run upfront. Then, based on different indications and the results obtained from the first round of tests, other molecular pathology tests such as PCR, FISH, microarray, and NGS will be performed as reflex. The focus of our project and this presentation is chromosome genomic array testing, CGAT, or chromosome microarray testing, CMA, and next generation sequencing, NGS. These two tests have been incorporated as part of the standard of care by many health institutions around the country in recent years. Despite that, the laboratory professionals are excited about the clinical utilities of these two tests. One big question remains: are we actually getting paid? So medical reimbursement is very complex. The decision on whether or not a test will be paid depends on many factors, like the current procedural terminology or CPT code that are used to build these tests, the fee schedule published by Center of Medicare and Medicaid Services, CMS, and also the pre-negotiated coverage and rate between each institution and different insurance companies. Dr. Bosler covered much of this topic back in the previous talk. For these two tests, the American Medical Association, AMA, introduced new CPT code in January of 2015. Unlike most single genes assay with Tier 1 CPT codes and got priced first, these two tests were assigned Tier 2 codes and were priced later. Overall, there is a scarcity in information. So to better understand the reimbursement landscape of these novel technologies in the clinical realm, we decided to perform a comprehensive analysis using our own institutional data. So these are the testing platforms used at our institution. For CGAT, we use the Cytoscan HD platform, and for NGS, the primary panel we use is the hotspot for targeted mutation detection. The genes included in this panel is shown on this slide. We look at all patients who received CGAT and NGS testing in the fiscal year between July 2016 and June 2017. There were 284 CGAT studies and 272 NGS studies. We worked with SCCA Corporate Finance to obtain data on CPT code-based reimbursement. For CGAT, we look at all studies built with CPT code 81406, a molecular pathology procedural code that covers a huge range of single-gene DNA sequence analysis and includes the cytogenomic microarray analysis testing for copy number abnormalities and loss of heterozygosity using single nucleotide polymorphism or SNP technology. A1229 is another code that can be used to build CGAT, but this code was not used during the time frame of which our data was obtained. However, don't completely forget about this code because I'll talk about it later. For NGS, we look at studies built with CPT code 81450 and 81455. Both codes are for targeted genomic sequence analyses for neoplasia. The main difference is that the former is for 5 to 50 genes and the latter is for 51 or more genes. Before I dive into the actual data, I would like to first introduce the terminology I'll be using for the rest of this presentation. First, appropriately paid is defined as cases which our institution received the expected amount of reimbursement. For public insurance, this amount is based on the fee schedule determined by CMS, whereas with private payers, this amount is based on the contract between the institution and the insurance companies. Written off, on the other hand, is defined as cases which our institution did not receive any payment. This may be because the payer doesn't cover the test, or many other reasons or circumstances that result in the reimbursement request being denied. As I'm not a biller, I'm not going to go into details on why a study is written off. Last but not least, all data that is presented here is expressed in percentage of cases, but not in dollar value. So this first slide compares the overall reimbursement between CGAT and NGS. Of all the cases we looked at, we receive appropriate payment for approximately half of the CGAT cases. NGS has a higher proportion of cases getting proper reimbursement, with over 3/4 of cases. The reason behind why a higher percentage of NGS cases receive appropriate payment is due to the payer mix of patients we receive at our institution, and also the insurance coverage decision for these payers. Because these data are expressed in a percentage of cases, it does not mean that NGS brings in more money or that NGS is more profitable than CGAT overall. Now let's look at the payer breakdown of our institution. As shown on this slide, the payer breakdown of patients receiving both tests is quite similar. Around 45% of cases are being paid by commercial insurance companies, and around half of the cases are being paid by public insurance, which includes Medicare and Medicaid. These numbers should not be very surprising, as this payer mix is observed in many institutions around the country. The rest of the patients are either self-paid or covered by other payers like TRICARE or VA. As more than 90% of our cases are commercial, Medicare, or Medicaid, we focus our analyses on these primary payer categories. This slide shows a side-by-side comparison of cases that receive appropriate payment as broken down by the payers. When we look at the patients that were covered by commercial payers, we observed that for both CGAT and NGS, we receive appropriate reimbursement for around 60% of the cases. For public health insurance payers, this is where we saw the most drastic difference in coverage between CGAT and NGS. With Medicare, all 114 NGS cases were appropriately paid, whereas for CGAT, only 40% of cases were paid. A similar trend is also observed in Medicaid cases. This is because before this calendar year, Medicare did not have a price for CGAT, and Washington State Medicaid did not allow for CGAT. Now you may ask, if CGAT is not allowed or covered, then how come some cases actually got paid? We looked into the detailed billing data for some of these cases and found that some local administrators for Medicare and Medicaid actually paid for these cases on a one-off basis. Some of these cases were paid by charitable organizations or the Washington State-funded AppleCare. So as someone whose focus is on the technical aspect of the lab, I've always wondered if insurance company would be more likely to pay for a case with abnormal results, but decline payment for a case that has normal results. So we looked into the percentage of cases with normal or abnormal results that received appropriate reimbursement. As shown on this slide, for both CMA and NGS, normal and abnormal cases are getting reimbursed at a similar percentage. NGS cases with variants of uncertain significance appear to be written off at a higher percentage. However, there were only 12 cases in this category, which represent less than 10% of the total cases. Thus, we do not think this reduction in appropriately reimbursed case number is significant. After we determine that normal and abnormal results do not affect the reimbursement decision, we look into whether or not the disease indication affects the reimbursement decision. This slide shows the breakdown of different disease pathways we use for our institution. For both CGAT and NGS, about a quarter of cases are acute leukemias. This number includes cases of both lymphoid and myeloid lineage. For CGAT, there's another quarter of cases that are plasma cell neoplasm. These cases are currently not being tested with NGS, though this will change in the near future. The next major disease group for CGAT and NGS testing are myelodysplasia syndrome, MDS, and myeloid proliferative neoplasm, MPN. So we decided to focus our analyses on these 4 disease pathways. So this slide shows a comparison between the cases that have been appropriately paid and written off in different disease pathways for both CGAT and NGS. Overall, there is a higher number of cases getting appropriately paid in NGS. When we focus our analysis on PCN, AL, MDS, and MPN, we notice no significant difference in cases getting appropriately reimbursed. This suggests that when an institution has established the medical necessity to perform NGS or CGAT, insurance company generally would not challenge it. The eval HD pathway is how we categorize patients at our institution based on disease indications. However, from a billing point of view, the insurance company put emphasis on the ICD-10 code. So we did a similar comparison using the most commonly used ICD-10 codes. I don't expect any of you to read all the coding details on this slide, but the trend of reimbursement percentage you can appreciate is that it's actually quite comparable to the previous slide. Overall, there's a higher percentage of NGS cases that are appropriately paid, and no one code stood out as being better or worse than the other. In conclusion, overall NGS is being reimbursed at a higher rate than CGAT and CMA. This is mainly due to Medicare and Washington State Medicaid coverage decision. We didn't observe any significant difference in the percentage of appropriately reimbursed cases when we compare normal versus abnormal results, or when we compare clinical indications using diseases listed by pathway or ICD-10 code. So what's next? So first, CMS released price for CGAT in the fee schedule for 2018. This means Medicare and Medicaid will start paying for these tests. This is great news considering CGAT has been offered for several years and labs have been performing the test without knowing whether or not we'll get paid. And the second point is, as Dr. Bosler mentioned, briefly covered in the previous presentation, is regarding the National Coverage Determination for NGS that was released in CMS several months ago. The big win that he described in that presentation was incorporated into the final determination after CMS received more than 300 comments from our community. So this speaks to the power of advocacy. So where to go from there? As you can see, the newly released fee schedule for the CPT code for 81229 is for $1,500, whereas for 81406 which is the code that is used by many labs right now for oncology array testing, it is only $280, which is way below cost. Although most labs have been using 81229 for constitutional array testing and 81406 for oncology array testing, are they really the most appropriate mapping? Should we as a community advocate for higher reimbursement for 81406, or should we advocate for better mapping for the neoplasia CGAT 81229? As Dr. Lennon stated this morning at the opening, CGC has already submitted a petition to remapping the genome-wide copy number and SNP array in the setting of neoplasia testing to CPT code 81229. So very soon, AMA is going to come to us and ask for public comments. I would strongly encourage everyone to put in the comments, because as we learned from the success story of the NGS reimbursement, we really can make a difference in situations like this. So it will really help our community, and kudos to the CGC board for doing this. I would like to thank Dr. Fong for all her support and guidance throughout this project. And I would also like to thank University of Washington Hematopathology Lab, SCCA Cytogenetics Lab, and also SCCA Corporate Finance for the collaboration on this project.
Thank you, Melissa. I think we have time for one question.
Thank you for sharing the data. Can you comment on the level of variability in terms of reimbursement amount from one payer to the next?
Unfortunately, no. I got very high-level data from our corporate finance, and it's interesting how variable the data can be. And I remember asking, because it's really funny you brought that up, because originally there is some data, and I map it out on a diagram, and I show it to them, and they were like, wait, something is wrong. So they went back and revisited their data and gave me a new set. So I would like to say, no, I don't know how to explain some of the variability.
Okay, but you do see great level of variability.
Yes,
is what you're
totally,
saying.
totally
Okay.
great variability.
Okay.
I actually have another slide, but it's not that. It's practically repeat testing within the year. You can actually, I have patients that got paid the first time and not get paid the second time, and vice versa. So it's a lot of variability.
Okay. And can you also comment on percentage of cases that are initially denied but eventually paid out?
That I cannot comment, sorry.
Thank you.
One more. Thanks for this comparison. This is really good collection of like live samples and gives people an idea of how it works. So on the subject of determining copy number variations by either array or next-gen, more and more people are going to do this by next-gen. Right now, if the assay looks for copy number variations, but is based entirely on next-gen, what happens? In terms of codes, how would you build that? And, and then is there an argument that people who do this, go into an extra effort to do this by next-gen, should be additionally paid because they essentially cover the copy number variant code?
That is a great question. I mean, I can only comment on what I have seen at my institution, and based on what I have found, is technology specific. So it doesn't really matter what how you use the technology. Like you can use the NGS to do copy number, but the problem is from our point of view is if you report it out as an NGS result, then you can charge the NGS code. While if you're using the array and you're reporting array data, then you can use the array code.
Yeah, just a follow-up comment to that, and that is technically the descriptor for the NGS codes should include copy number evaluation. I believe it's in the descriptor. And so most people outside of the MolDX program, you would be able to use that code. If you're in a MolDX program, you'd have to use 81479. The other thing, too, is that if you've got something that's entirely uniquely yours, then you go for a PLA code. The thing that I'm going to add one other comment that I mentioned to Min here was that with regards to the microarray testing, one other option that we have is to get a new code. Let's get a new code built for what that is, because I'm curious, how many people here are using 81406 to do microarray testing? A handful? Yeah, for neoplasia, yeah. I mean, it's really surprising that that's so low, but if there's enough of us doing it, if there's volume, we'll get a new molecular code.